Thank you for coming. I'm delighted to introduce Lidya Tarhan, who's visiting from Yale University, where she's an assistant professor in the Earth and Planetary Sciences Department, as well as an assistant curator in the Peabody Museum. She studies the relationship between animals and the environment using a combination of sedimentology, geochemistry, numerical modeling techniques, and other novel experiments in both the field and the laboratory. She received a BA from Amherst College in Geology and English, and an MS and PhD from the University of Riverside, studying with Mary Drozer. Is that correct? Her CV is jam-packed with all kinds of information. I will share that she was a NSF graduate research fellow, an NSF postdoc, and recently received the Donath Medal from GSA in 2021. And I also should mention that she's the postdoc advisor for one of our recent graduates, Julian Wang, who I understand is thriving at Yale. And I'm also pleased to report that we'll soon be co-authors together on a stable strontium isotope study that's been provisionally accepted, but we don't know for certain. So we'll just go with provisional for now. But anyway, we're delighted that you're here, and thank you so much for coming, and we look forward to your seminar. Thank you so much.
Thank you, Andy. Yeah, it's a great pleasure to be here today. So thanks for having me. And I'm really excited to share some of my group's recent work on the evolution and the biogeochemical impact of bioturbation. So bioturbating, or burrowing and sediment mixing animals, are one of our foremost examples today of ecosystem engineers, of organisms that substantially modify their surroundings, not only transiently, but also on timescales that are evolutionarily and geologically meaningful. And when we look to bioturbation in marine environments today, we see that bioturbators directly influence both the physical and the chemical properties of the seafloor. Their activities oxidize seafloor sediments and enhance rates of remineralization. And by mixing and churning sediments, they increase their fluid content, transforming on average the upper 10 centimeters of the seafloor into a homogenized mixed layer. And due in part to this potent combination of rheological and biogeochemical effects, bioturbators in turn strongly influence the structure of seafloor communities. And when we're considering the evolutionary history of bioturbation, some of the aspects that remain least well constrained is how did ancient bioturbators influence contemporaneous biogeochemical cycling and ecosystem structure? And what relationship, if any, is there between the evolutionary history of bioturbation and geobiological records of change in Earth's ancient oceans? And our predicted responses to these questions fall broadly into what we can think of as a couple of different camps. The first of these is a framework that's been informed largely by biogeochemical modeling, using which a number of recent studies have argued that the emergence of bioturbation would have had a deleterious impact upon contemporaneous ocean waters and may have driven both productivity crises and widespread episodes of ocean deoxygenation. And on the other hand, we have a framework that's been informed more by macroecological and macroevolutionary theory, which would predict that the emergence of bioturbation, like other forms of ecosystem engineering, should ultimately enhance the flow of nutrients through ecosystems, and thus mediating greater increases in organism size and body mass, as well as ecological complexity. And I've long been fascinated by the apparent dichotomy between these two different frameworks. Does this mean that one of these must be fundamentally flawed, or might there potentially be some way to reconcile them? But before we can really jump into understanding the biogeochemical or the ecological impacts of bioturbation, we need robust first-order records of how bioturbation has changed over our planet's history. So if we look to stratigraphic archives, for instance, when do we first start to see evidence for the development of a sedimentary mixed layer? And when do we see indications that seafloor sediments became mixed to the extents, to the intensities and the depths that characterize seafloor environments today? And this is a critical question because historically, the advent of bioturbation, as recorded by the trace fossil record of trackways and trails and burrows, so the record of how animals interact with sediments, have been invoked as the driver for a wide range of changes that we observe in terms of both geochemical and paleontological phenomena across the Neoproterozoic, the late Precambrian, and early Phanerozoic transition. such as declines in the abundance and diversity of microbial buildups, such as stromatolites, the disappearance of some of Earth's earliest complex animal ecosystems, like the enigmatic Ediacara biota, closures of windows of exceptional fossilization, as well as the rise of oxidants in Earth's oceans, and perhaps even the stabilization of Earth's long-term carbon cycle. But were early Phanerozoic, and in particular were early Paleozoic seafloor sediments, as well mixed as they're commonly assumed to have been on the basis of this trace fossil record? And to what extent do late Precambrian, do Neoproterozoic through lower Paleozoic geobiological records of change actually align with major inflections in either the intensity or the extent of bioturbation? And historically, this has been something of a thorny issue to unravel because there's been a great deal of reliance upon that trace fossil record. And in particular, individual trace fossil specimens archived to museum collections, as well as observations of trace fossil assemblages represented in the ichnological or the trace fossil literature. And although trace fossils can be enormously informative in telling us about major biological innovations, major changes in animal behavior, they don't necessarily give us an accurate representation of average of background seafloor conditions. So for instance, in the case of museum specimens of trace fossils, these are commonly skewed towards the largest and the best preserved examples, and they're frequently archived without sedimentological context. Moreover, a number of the key studies that have argued for an early escalation in intensities of bioturbation, for saturation perhaps even as early as stage 2 of the Cambrian, with intensities and depths of bioturbation characteristic of the modern seafloor, have tended to rely heavily on maximum metrics of bioturbation, maximum depths of burrowing, and maximum intensities of sedimentary fabric disruption by burrowing. But maximum metrics of bioturbation aren't necessarily representative. And this is in fact very nicely demonstrated by Cambrian and Ordovician bioturbation data from carbonate-dominated shallow marine successions exposed in the Western US. And what I'm showing you here are 6 representative units that are part of a much larger data set that spans thousands of meters of stratigraphic section. And I've organized these with our 3 older units, so Cambrian age, so spanning about 541 to 485 million years ago in the upper row, and 3 of Ordovician age, so slightly younger, about 485 to 444 million years ago in the lower row. And what you see along the x-axis of each of these diagrams is a common metric for intensity of bioturbation that we call the ichnofabric index. And the ichnofabric index measures the extent to which bioturbation disrupts the original physical stratification with which sediments are deposited on the seafloor. So at the lower end of our spectrum, an ichnofabric index value of 1 corresponds to no bioturbation, to just purely that physically stratified, for instance, that laminated sedimentary fabric. Toward the upper end of that spectrum, an ichnofabric index value of 5 corresponds to bioturbation disruption so intensive that we no longer have any vestige of that original physical stratification. It's been entirely overprinted by burrowing. And in fact, their theoretical maximum value is an ichnofabric index value of 6, which corresponds to wholesale homogenization. So bioturbation activity so intensive that it is self-erasing. Not only do we have no vestige of that original physical fabric with which those sediments were laid down on the seafloor, but also we no longer see discrete burrows preserved. We only have a massive, a homogeneous sedimentary fabric. So essentially true mixed layer development. And so what we see in each of these 6 examples is that the maximum ichnofabric index value, which for all these units is 5, is far higher than the mean, than the average ichnofabric index value, which for these units ranges between 2 and 3. Moreover, the relative abundance of these higher ichnofabric index values is quite low, indicating that although we rarely see instances of more intensive fabric disruption, on average in these Cambrian and Ordovician carbonate-dominated shallow marine platform and shelfal settings, more limited extensive bioturbation, lower to intermediate ichnofabric index values are the norm. And this perhaps shouldn't surprise us when we consider the extent to which bioturbation varies on the seafloor today. And in fact, both the depth of the sedimentary mixed layer and intensities of sediment mixing vary considerably across the modern seafloor. So as an example of that, this is a global heat map of modern marine mixed layer depths, which we've generated using a machine learning framework from not only all currently available modern marine mixed layer depth data, but also using a random forest tree-based ensemble, we've been able to predict at a much finer spatial scale variability in mixed layer depths using environmental and ecological predictor parameters. So clearly, bioturbation intensities and depths are quite heterogeneous on the seafloor today. But to resolve any of these debates regarding either the biogeochemical or the paleontological impact of bioturbation, we need better first-order records of how bioturbation has varied across our planet's history, and in particular, how different background intensities and styles of bioturbation have changed over the course of the early Paleozoic, of the first 120 million years of the Phanerozoic, which is the interval that saw the emergence and the radiation of a wide range of different animal groups and body plans, but also the establishment of a range of more complex animal ecologies, with initially the Cambrian explosion and subsequently the Great Ordovician biodiversification event. So we know that from the trace fossil record, that we have vertical trace fossils. We have three-dimensional trace fossils recording animals penetrating into seafloor sediments that extends as far back as the beginning of the Cambrian and even the latest Precambrian, even the latest Ediacaran. But what about the sedimentological impact of bioturbation? How intensively or extensively mixed were those early Paleozoic seafloor settings? So field-based studies that have collected detailed and continuous sedimentological and paleontological records of sediment mixing in a stratigraphic context. And in particular, those studies that have prioritized collection of data that allow us to say something about the extent of sedimentary fabric disruption by bioturbation, or about the consistency of seafloor sediments at the ancient sediment-water interface, how cohesive and firm and poorly mixed these were, or alternatively, how fluidized, how soupy, how well bioturbated these were, have started to paint a very different picture of early Paleozoic bioturbation than what has historically been envisioned. So for instance, heterolithic siliciclastic stratigraphic records, so thinly intercalated mudstones, siltstones, and sandstones, which represent a wide range of shallow marine and continental margin settings. Data from these successions indicate that although animals clearly began to colonize seafloor sediments at a relatively early stage in their evolutionary history, the development of extensively mixed, homogenized sediments, true mixed layer development, was a much more protracted process. And that even by the close of the early Paleozoic, some 120 million years after the Precambrian Phanerozoic transition, the depth of the sedimentary mixed layer, although gradually increasing over this interval, remains substantially shallower than what is characteristic of the mixed layer in modern oceans. But given the variability we see in bioturbation intensities and depths in the modern oceans, what can we say with confidence about variability of bioturbation in the Paleozoic oceans? Can we actually discern major paleoenvironmental trends in the evolutionary history of bioturbation? Or can we even resolve such fundamental but long-standing questions, such as whether bioturbation ramped up more rapidly in more onshore and shallower water or more offshore settings? So in order to pursue this question, my colleagues and I have recently begun working in the Cambrian and Ordovician portaport succession of southwestern Newfoundland, which is a succession that is mixed carbonate siliciclastic and contains a number of units of this key Cambrian and Ordovician age, encompassing these radiations and animal diversity as well as ecological complexity. Moreover, these successions are beautifully exposed in sea cliffs along the southwestern Newfoundland coastline. And importantly, these Cambrian and Ordovician units also encompass a range of facies representing different depositional environments, both more onshore shallow water settings and more offshore environments. So working with the port-to-port succession allows us to make tractable the question of not only how has bioturbation changed through time, but to what extent did it vary across depositional environments through this key interval. And what we found after measuring about 400 meters of stratigraphic section at a detailed scale, at the centimeter scale at which previous studies have indicated bioturbation actually varies, is that there is considerable variability in intensities of bioturbation throughout the port-to-port succession. And although we occasionally observe intervals characterized by more intensive bioturbation, for instance, ichnofabric index values as high as 5, so total disruption of physical sedimentary fabrics by bioturbation, those tend to be quite rare. On average, we see that levels of bioturbation throughout the port-to-port succession are lower to more moderately developed. And in fact, average ichnofabric index throughout the succession is less than 3, so only moderate intensities of sediment disruption. However, we do see that those facies recording the most near shore, the shallowest water successions, for instance, beach and shore face environments, are characterized by higher intensities of bioturbation at a statistically significant level. So that allows us to answer this long-standing question with data from the port-to-port succession indicating that bioturbation did ramp up more rapidly in those most near-shore settings. However, what we see from this succession, as we saw in heterolithic siliciclastics worldwide, is indication that across a wide range of early Paleozoic seafloor successions, levels of bioturbation remained more limited, only moderately developed throughout this interval. So you might recall that one of the observations with which I began concerned the extent to which modern bioturbators impact the rheology, the consistency and the erodibility of modern seafloor sediments. So can we say anything about the extent to which ancient bioturbators may have similarly impacted the rheology of the early Paleozoic seafloor? And to tackle that question, we can turn to soil marks. So soil marks are current-mediated deformational and erosional structures. They are formed typically either by sediment-laden fluids or a fluid-propelled tool impinging upon and gouging seafloor sediments. And they're perhaps most familiar to us as paleocurrent indicators because they form in alignment with paleocurrents in a wide range of aqueous settings. And we, of course, call them soil marks because these gouges are subsequently filled in by sediments with the next sedimentary depositional event. And after the passing of geologic time, when these beds have been parted, we see them beautifully preserved as casts, as positive relief features on the bases of the overlying beds. So although soil marks are perhaps more familiar as a paleocurrent indicator, they can also be excellent paleorheological indicators. And this is because a large body of experimental evidence, as well as observations from natural settings and theoretical constraints, have indicated that these features will only form in cohesive or hydroplastic sediments. In other words, they will not form in soupy, well-bioturbated sediments. So using soil marks as a proxy for seafloor rheology, and specifically for the presence of hydroplastic or cohesive sediments, can we say something about how the rheology of the seafloor may have been shaped by bioturbation over the past 550 million years of the Phanerozoic? And in fact, it turns out that if we look to the abundance of these structures, or to the abundance of shallow marine successions, particularly tempestite-dominated successions recording these structures, that the abundance of those shallow marine records is distinctly non-uniform over the course of the Phanerozoic. And we see that the abundance of those units, of these features, is much higher in the early Paleozoic, in the Cambrian, Ordovician, and Silurian, and we see a subsequent decline over the remainder of the Phanerozoic. And this is robust regardless of whether we normalize to outcrop area, to sedimentary rock mass or volume, or to the duration of our various time beds. And this decline, this elevated abundance and subsequent decline, very nicely inversely mirrors what other independently proxies have told us is an interval of protracted development in bioturbation and of well-fluidized mixed grounds through this interval. So returning to some of those biogeochemical questions with which we began, you might recall we have these apparently dichotomous frameworks for what we might expect to be the impact of early Paleozoic bioturbation upon the global oceans. And on the one hand, we have expectations that have been guided by biogeochemical modeling, a number of studies of which have suggested that bioturbation would have initiated productivity crises and widespread episodes of ocean deoxygenation. And on the other hand, we have a framework that's been informed more closely by macroecological and macroevolutionary theory, which would predict that the emergence of bioturbation, like other forms of ecosystem engineering, should ultimately enhance the flow of nutrients through ecosystems and thus sustain greater organism size and ecological complexity. And in order to resolve this apparent dichotomy, what we really need to understand are what are some of the assumptions that are inherent to each of these frameworks. So to start with the first of these, recent efforts using biogeochemical modeling to explore the impact of early Paleozoic bioturbation upon contemporaneous biogeochemical cycling, and in particular upon the coupled phosphorus, carbon, and oxygen cycle, have tended to simplify phosphorus cycling to essentially a bimodal system, whereby in anoxic settings, phosphorus is preferentially liberated from organic matter via anaerobic degradation and recycled back up to the overlying ocean water. And in contrast, in oxic and bioturbated sediments, phosphorus is expected to be preferentially buried via absorption to iron oxides. And using this simpler framework for the phosphorus cycle, a number of studies have argued that early Paleozoic bioturbation would have driven decreases in organic matter burial, increases in phosphorus burial, and thus potentially throttled productivity and enhanced ocean anoxia at various intervals of the early Paleozoic. However, this more simplified framework for phosphorus cycling and bioturbation's impact upon the phosphorus cycle actually excludes the chief sink through which phosphorus is buried in the oceans today. And that is as authigenic phosphorus, or carbonate fluoroapatite, which I'll hereafter refer to as CFA. And CFA is actually the chief phosphorus burial sink, not only along continental margins experiencing upwelling, as we might expect, but also in a wide range of other marine settings, such as passive margins, as well as the deep sea. However, authigenic phosphorus, CFA, is typically not included in biogeochemical models because including it does require greater complexity. For instance, CFA formation is dependent upon CFA saturation state, which is impacted by factors such as pore water pH and other aspects of the carbonic acid system. Additionally, studies that have used biogeochemical modeling to explore the impact of early Paleozoic bioturbation upon contemporaneous biogeochemical cycling have tended to simplify bioturbation to a single term. However, bioturbation really isn't a single process. It's an umbrella that includes a much broader range of animal behaviors and animal sediment interactions, some of which we can loosely group together under the umbrella of biodiffusion, which is the randomized movement of sedimentary particles, so essentially mixed layer development. And we can represent biodiffusion with an intensity coefficient, dB, as well as with an attenuation coefficient, or essentially a depth, xbt. Others of these processes we can loosely group together under the term bioirrigation, which is the non-local movement of pore water solutes, for instance, via open burrows. And as we do for biodiffusion, we can represent bioirrigation with an intensity coefficient, in this case alpha, as well as with an attenuation coefficient, xbi. And although it's been traditional in using geochemical modeling to explore the impact of deep time bioturbation, to simplify representations of each of these processes, I would argue that if we really want to understand the mechanisms that underpin these interactions, we need more realistic representation of each of these aspects. So toward that end, over the past 2 years, my colleagues and I have developed a new 1D diagenetic model, or sedimentary reactive transport model, which we've termed SEDChem. And SEDChem, like all diagenetic models, is built upon Bob Berner's simple diagenetic equations, which you see here, and which changes in the concentration of either solutes or solid phase species over time and with depth in the sediment pile are a function of not only chemical reactions, but also transport processes. And SEDChem includes over 40 such chemical reactions, including a full suite of microbially mediated redox reactions, as well as carbonate chemistry, including CFA formation. And it also includes multiple styles of bioturbation, both biodiffusion and bioirrigation. In using SEDChem, we can closely replicate empirical sedimentary geochemical data, both dissolved species and solid phase species from a variety of well-characterized modern marine sites, such as the particularly well-characterized Friends of Anoxic Mud or foam site in Long Island Sound, which is just offshore of Guilford, Connecticut, and which is a site from which both phosphorus speciation and bioturbation data have been collected. However, we can also, using SEDChem, closely replicate, without need for any additional parameterization, data from other well-characterized marine sites, both other continental margin settings, as well as sites in the deep sea. And we've been using the SEDChem model to explore the relationship between different styles and intensities of bioturbation and phosphorus cycling. So what you see here in this slide are 4 different panels, each of which represents one of those 4 bioturbation parameters that I introduced previously. And I've organized this with the biodiffusion parameters, our biodiffusion intensity coefficient, dB, as well as our biodiffusion attenuation coefficient, so essentially our depth, xbt, on the left, and our bioirrigation parameters, including our bioirrigation intensity coefficient, alpha, as well as our bioirrigation attenuation coefficient, xbi, on the right. And in each of these cases, these are represented fractionally, with 1 or 100% representing the values that we observe at that foam site. And these are sensitivity analyses, so in each of these cases, all other parameters, both bioturbation and biogeochemical parameters, were held constant as each of these were varied individually. And along the y-axis, you see a couple of different representations for phosphorus burial. The solid lines denote CFA, so our authigenic phosphorus burial flux. The dashed lines indicate total phosphorus burial efficiency. So that's the balance between inputs of phosphorus to the system and outputs in a variety of burial terms, including not only our authigenic phosphorus, but also organic bound phosphorus and iron oxide sorbed phosphorus. And I've shown each of these so that you can see that changes in total phosphorus burial efficiency closely mimic and in fact are driven by changes in authigenic phosphorus burial. And lastly, the different colors that you see in these plots correspond to different maximum XVI values, so different depths of bioirrigation, with the green curves corresponding to more deeply bioirrigated systems and the blue curves to more shallowly, to more intermediately bioirrigated systems. So stepping back to a first order, we can see that bioturbation has a complex, frequently nonlinear, in some cases even non-unidirectional relationship with phosphorus burial. And biodiffusion and bioirrigation actually appear to have competing effects upon phosphorus burial, which is something that hadn't previously been noted. Increases in biodiffusion can lead to increases in phosphorus burial. And this is because biodiffusive sediment mixing brings sedimentary particles, including phosphorus particles, deeper into the sediment pile, into the more anaerobic regions of the sediment pile, where it may be easier under the higher pore water pH values characteristic of those zones to achieve saturation with CFA. And also at those greater depths, it's more challenging for any phosphate liberated from those phases to be effluxed back to overlying bottom waters. In contrast, increases in bioirrigation lead to decreases in phosphorus burial. And this is because although bioirrigation has the potential to enhance the formation of iron oxides, and thus the potential for absorption of phosphorus to those oxides where it could theoretically undergo sink switching at depth, it also increases the relative importance of aerobic degradation, making it more challenging to achieve saturation with CFA, which again typically requires higher pore water pH values. Recent studies have also suggested that the emergence of bioturbation may have operated as a stabilizing or a negative feedback on Earth's carbon and oxygen cycle. So in order to test this hypothesis, we've also been using the SEDChem model to explore the impact of not only changes in bioturbation, but those changes in bioturbation coupled with changes in environmental factors that have been previously linked closely to the phosphorus cycle upon total phosphorus burial efficiency. For instance, bottom water oxygen concentrations, which you see here in the plot on the left, as well as rates of organic carbon delivery to the seafloor, or JOC, which you see over here in the plot on the right. And as in the previous set of sensitivity analyses, we also explored these feedbacks under different XVI values, so different depths of bioirrigation. And as in the previous slide, the green curves here correspond to more deeply bioirrigated systems. The blue curves correspond to more intermediate, shallower depths of bioirrigation. And here the black curves correspond to a system without any bioturbation. And what we see is that if we were to mimic an environmental perturbation, for instance, a decrease in bottom water oxygen concentrations under intermediate depths of bioirrigation, so here following this blue curve on the left from right to left, we see a decrease in phosphorus burial efficiency, corresponding to an increase in recycling of phosphorus back to overlying ocean waters, where it could fuel greater productivity, and also lead to a greater potential for local anoxia, which would push us further to the left down this curve. So further and further down that positive feedback, that destabilizing loop. And concurrently, if this does lead to increases in productivity, or even just to preservation of organic matter, this could also drive increases in JOC, increases in rates of organic carbon delivery to the seafloor, which likewise in a more intermediate bioirrigation scenario would drive us towards lower phosphorus burial efficiency, enhanced phosphorus recycling, and the potential for increased productivity, but also increased local anoxia. So in each case, this operates as a positive or destabilizing feedback. However, on geologic, on million-year time scales, we might expect that this would ultimately operate as a negative, as a stabilizing feedback. And that if increases in productivity or preservation led to increases in organic matter burial, that would ultimately drive increases in ocean and atmospheric oxygen levels. Intriguingly, however, in more deeply bioirrigated systems, those represented by the green curves, we see that this can operate as a stabilizing, as a buffering feedback against environmental perturbation in the case of JOC. However, in the case of bottom water oxygen, the effect of a perturbation under more deeply bioirrigated conditions is indistinguishable from a system without any bioturbation, as represented by the black curves. So if we return to our stratigraphic archives and recall that these indicate that although animals clearly began to colonize a wide variety of shallow marine seafloor settings quite early in their evolutionary history, as recorded by the trace fossil record, the development of extensively mixed, homogenized sediments, true mixed layer development, was a much more protracted process. This might suggest that in our diagenetic modeling framework, that many early Paleozoic shallow marine settings were ones in which bioturbation was dominated by bioirrigation, by burrowing, and in which biodiffusive sediment mixing was relatively muted. And this might indicate that in those early Paleozoic shallow marine settings, increases in bioturbation, that is, increases in bioirrigation, would have actually led to decreases in phosphorus burial, in contrast to the strictly positive relationship that has been previously proposed. Intriguingly, however, subsequent increases that we might expect in biodiffusion in more deeply bioirrigated systems, represented by the green curves, actually have almost no impact upon phosphorus burial efficiency. And in fact, phosphorus burial efficiency in those more deeply bioirrigated systems is indistinguishable from that in modern settings today that have no bioturbation, as indicated by the yellow circles. So clearly, bioturbation has a more complex, less straightforward relationship with phosphorus cycling than has perhaps historically been envisioned. So one of the other things that we've been using the SEDChem model for is to explore the influence of a wide range of different boundary conditions, and not bioturbation alone, upon phosphorus burial. So what you're looking at here are the aggregated results of a whole series of sensitivity analyses exploring the impact of each of the boundary conditions that you see noted here along the horizontal axis. And that includes bottom water concentrations of a variety of solutes such as phosphate, sulfate, magnesium, oxygen, and calcium, as well as bottom water pH values, and also bioturbation, as well as rates of organic carbon delivery to the seafloor. And in each of these cases, we've modulated these factors individually, again, in a sensitivity analysis framework, but only within the range in which we observe those factors to vary in the oceans today, or for which we have empirical proxy data indicating that they vary to this extent in the recent geologic past. And the horizontal line that you see here represents baseline values as indicated by the values we have at the foam site in Long Island Sound. And what you can see is that there are actually a variety of boundary conditions, including bioturbation, that can act as strong levers upon the magnitude of phosphorus burial, which is here represented by the concentration of CFA, of our autogenic phosphorus burial term. So bioturbation is among these, but in fact, there are a number of other boundary conditions that are equally strong, if not stronger, levers upon changes in phosphorus burial, such as bottom water oxygen concentrations, rates of organic carbon delivery to the seafloor, and in particular, bottom water calcium concentrations. And mechanistically, this can have a large impact upon phosphorus by influencing the saturation state of carbonate fluoroapatite of CFA. And when we consider the magnitude to which these changes in seawater calcium concentrations can act as a lever upon phosphorus burial, this is really potentially quite exciting because we know that seawater calcium concentrations have varied significantly over the past 500 or so million years of the Phanerozoic. And this might suggest that calcium, although perhaps historically underappreciated as a lever upon phosphorus cycling, may have modulated major changes in phosphorus burial and thus an Earth's long-term carbon and oxygen cycle. But first, a little bit delve more empirically into the recent geologic past to test the robustness of this framework. If we look to sedimentary geochemical data from the past 80 million years, for instance, ODP data from a variety of ocean basins, we see that not only is CFA the major phosphorus burial sink in these archives, but also that changes in CFA and in total phosphorus burial do indeed appear to be tightly coupled to changes in seawater calcium concentrations across this interval. And although there remains substantial debate about what exactly drives or most strongly drives these changes in seawater calcium or seawater magnesium calcium ratios, whether, for instance, they're driven more strongly by supercontinental scale cycling in extensive seafloor spreading and thus exchange between seawater magnesium and basaltic calcium, or whether, for instance, changes in temperature by modulating the extent of magnesium-rich clay formation in off-axis settings may have been a stronger lever upon these concentrations. Regardless of the driver, the records themselves are well established and supported by multiple proxies. So in light of these Cenozoic and late Mesozoic data and the indications from our sensitivity analyses using SEDChem, we can couple SEDChem to a simple carbon-oxygen ocean atmosphere model, which borrows elements from both Tim Lenton's COPS model, as well as, for those of you who are familiar with it, the Van Capellen and Engel model, and additionally takes constraints from the geologic archives for not only changes in factors such as seawater calcium concentrations and bottom water oxygen concentrations, but also in extensive bioturbation in order to explore what the impact would be over longer geologic timescales upon marine and atmospheric oxygen levels. And what we observe is that increases in seawater calcium concentration not only can drive increases in phosphorus burial, but that these in turn can directly drive decreases in oxygen concentrations in the ocean and atmosphere system, suggesting that seawater calcium concentrations as a lever upon diagenetic phosphorus cycling can indeed be a major driver of secular changes in the ocean atmosphere oxygen cycle over geologic timescales. So starting to summarize some of what we've learned through this process, I think that we have robust evidence from a variety of sedimentary, stratigraphic, and paleontological archives indicating that the development of bioturbation was a protracted process across a wide range of early Paleozoic seafloor settings. And in my mind, one of the most intriguing aspects to emerge from this work is the substantial lag we see in time between the appearance of organisms that clearly were capable of colonizing seafloor sediments, as recorded by the trace fossil record, which extends back to even the late Ediacaran, and the development of widespread, well-mixed seafloor sediments. So this is a pretty compelling example of what in paleontology we would call a macroevolutionary lag. And this is a lag that not only encompasses the major radiations in animal diversity and body plans of the Cambrian explosion, but also the major increases in animal ecological complexity and establishment of new ecological niches as part of the Great Ordovician biodiversification event. So what might be the potential drivers of this substantial lag? One possibility that we have to perhaps consider is whether this lag could relate to a delay in the appearance of the necessary physiological toolkit among bioturbators, either at the anatomical level or even at the genomic level. However, again, we have a trace fossil record that extends all the way back to the beginning of the Cambrian and the late Precambrian. Moreover, although rare, we do see instances of more intensive sediment disruption, for instance, of ichnofabric index values as high as 5. So I think that this possibility of a delay in the toolkit holding back bioturbation is a less likely scenario. So if not a delay in the emergence of physiological ability, then there must be some other factor, either another evolutionary factor we haven't accounted for, or perhaps an ecological or an environmental factor. And one intriguing possibility rests with the work of Charles Thayer, who in the late 70s and early 80s suggested that the middle to later Paleozoic may have been a time of major radiation in what he called the biological bulldozers. And biological bulldozers are apex bioturbators today. They are the most efficient bioturbators, in part because they are mobile deposit feeders, like this holothurian or sea cucumber. So not only because they're mobile, do they impact a tremendous volume of sediments relative to other infaunal or burrowing animals, but they also chemically interact with those sediments because they're deposit feeding, they're ingesting those sediments. And Thayer based his hypothesis of a middle late Paleozoic radiation in the bulldozers upon not only the rather sparse body fossil records of clades of organisms that today contain major biological bulldozers, but also upon declines in the diversity of what he called the ISOS, the immobile soft sediment dwelling suspension feeding organisms that would have been particularly vulnerable to the destabilization of sediments and increases in seafloor erodibility and of bottom water cloudiness that may have followed in the wake of a major radiation of biological bulldozers. Another possibility has to do with ecological feedbacks with substrates. So given our evidence from the Solmark record for the persistence of cohesive and hydroplastic sediments through the early portions of the Paleozoic, and given how metabolically demanding activities like bioturbation are, perhaps what was needed was the rise of organisms like the bulldozers to essentially act as icebreakers to help break up those cohesive seafloor sediments and make them more passable to smaller or less efficient bioturbators. And perhaps there's also a role for environmental factors in this secular evolution of bioturbation. For instance, there's a large and growing body of evidence indicating that the low dissolved oxygen concentrations that appear to have been characteristic of much of the Neoproterozoic may have persisted a considerable ways into the Paleozoic. So again, considering the metabolically demanding nature of activities such as bioturbation, perhaps low dissolved oxygen levels, particularly if coupled with high seawater temperatures, as have been reconstructed for the early Paleozoic, may have acted to keep bioturbation largely in check, except in rare circumstances. So hopefully the other thing that I convinced you of today is that early Paleozoic bioturbation cannot be simplistically invoked as the primary lever on changes in contemporaneous phosphorus and carbon and oxygen cycling. And in fact, early Paleozoic bioturbation, which was likely bioirrigation dominated, may have actually enhanced phosphorus cycling and spurred greater productivity, in contrast to what previous studies have suggested, but bringing us more in line with the expectations of macroecological and macroevolutionary theory, that the emergence of key ecosystem engineering behaviors like bioturbation should ultimately enhance the flow of nutrients through ecosystems and thus spur greater productivity and ecological complexity. However, at least initially, bioturbation may have in fact acted as a destabilizing feedback on biogeochemical cycles experiencing environmental perturbation. And lastly, when we're interrogating questions about the coevolution of biogeochemical cycling and bioturbation through Earth's history, given that the net effect and even the directionality of these interactions may not be intuitive, I think this reinforces that not only do we need very robust first-order records of how bioturbation has changed through time, but we also need to examine these interactions in a more mechanistic modeling framework. Now, with that, I'd like to thank a number of colleagues who were instrumental to this work, as well as a variety of sources of funding for their support. And thank you again for the invitation to speak here today, and I'm happy to take any questions. Maggie?
Really cool, really cool. I'm a big fan of this sort of more nuanced modeling approach. We're learning a lot that way, and I just learned a lot right now. I'm really, obviously, that tipping point in the mixed layer depth is truly fascinating. Having, you know, your end members, and so I'm wondering about the sensitivity of that, you know, how, so 0 to 0.8 is obviously a cliff, and then 0.8 to 2 is a different cliff, but what does that curve look like? And have you tested that? Obviously, the calcium concentration question is incredibly important to all of our modeling, but how do those co-vary? So in a low calcium universe, for instance, is 0.8 still the tipping point, or does that move around? And so could you imagine sort of an interplay between those changes occurring on relatively long time scales that lead to sort of these larger scale secular trends?
Yeah, that's a great question. So we haven't modeled the full combination of changes in calcium that we might expect based upon the fluid inclusion records, for instance, for the Phanerozoic, but the full range of bioirrigation depths that we might anticipate. But we have taken a little bit of a similar approach with biodiffusion, and we see that basically at higher calcium, it just intensifies the sensitivity. But that is a good question in terms of bioirrigation, and I don't think that we've quite explored where those thresholds are in bioirrigation, whether or not that attenuation shifts to, you know, to 1 or to 1.2 as being a major threshold under, say, you know, Cambrian calcium concentrations. So yeah, that would be a really interesting avenue for future work to try to tease this apart in more than sort of the sensitivity framework that we've done here. Andy?
Yeah, I'm going to follow up questions to that. First, thank you for visiting. Thank you for the seminar, which I really enjoyed. In your model, the diagenetic model of the biodiffusion term, so this, can you explain that, how that parameter works? I know you sent that paper to us for lab group meeting, and we spent some time looking at it, and I'm just not clear how that's parameterized, the A term, and then there was a couple terms in parentheses. Yes. Here. So how does that work in the context of the model? Like how do you actually numerically parameterize this?
Yeah, so in the baseline conditions that we used for this particular modeling exercise came from those foam values that previous studies have determined. In biodiffusion intensity coefficient, so this DB term, that has historically been reconstructed using a variety of different proxies. Some of those involve radio tracers, and some of the same proxies are also used to approximate mixed layer depth. So for instance, people commonly use thorium-234 or lead-210 or radium-226 to track rates of turnover in the sedimentary profile. Some folks use sort of more textural recognition, which is perhaps more analogous to what we can do in the geologic record for many of these successions, and look at fabric disruption using, for instance, X-ray techniques and looking at X-radiographs of the modern seafloor. Some folks have used things like chlorophyll A or tektite beads. So there have been a variety of tracers that have been used, particulate tracers that have been used, both radiometric and not, to try to untangle that for the modern ocean. And then the alpha, the bioirrigation parameter up here, that is often parameterized using the profiles of iron and manganese in the upper portion of the sediment pile in that dissolved phase. So it's typically done from modeling the empirical data that we get from the modern marine seafloor.
So I had one additional, but I had one additional question.
Yeah.
Down in the weeds, but on your plot of the SOMARs, was that one or?
Yeah.
Through time, what was going on in the Permian? Like,
Oh, I
like,
know, that's everybody asks that, and it's like a good question.
all my own.
And I would like to know more as well. It is a little bit puzzling. So this is normalized to outcrop area. So, we could make arguments about sort of supercontinent assembly and breakup, or, the prevalence of marine Permian successions, but it is normalized. So that was my first thought, but it is normalized. So it's hard to sort of pigeonhole it into that category as being an artifact of abundance of the sedimentary rock record for the oceans as a whole. So, one possibility is that there really is much more intensive bioturbation over that interval, but if so, it's pretty striking. It's the increase in the Triassic is less than I might expect in the wake of things like the PT mass extinction. A number of people have documented that there are increases in trace fossil preservational fidelity, including things that are formed at the sediment water interface, as well as features like soil marks, and also things like a resurgence in microbially mediated sedimentary structures. I think probably a part of this had to do with the sort of level of trade-offs between binning and time constraints for these predominantly siliciclastic and sandy units. So if I had been able to bin at a finer level, perhaps we would have seen the Triassic shoot up more.
Yeah.
But at the period level, it's sort of more in line. So that does sort of setting aside the Triassic, which I'm sure some portion of that, at least in the lower Triassic, is a sort of response to extinction. And decreased abundance and decreased activity of bioturbators. This is a really striking decrease that's sort of not paralleled until we get to the Paleogene and the Neogene. So yeah, I am myself a little bit intrigued, but also I don't have a sort of conclusive answer yet of why it is that the Permian is so low relative to, for instance, the Jurassic. So I currently have a student who's working on documenting, who's just starting out on a project documenting increases in mixing intensity through the Jurassic, with the idea that things like the marine Mesozoic revolution, we would expect to be a major increase in bioturbation if that had not already happened by the middle to late Paleozoic. So why it is that this is as high as it is also another question. So I think that, one of the things that ultimately I'd like to do is have more data for these younger intervals. There's actually not all that much data out there that has constrained intensities and depths of sediment mixing at the sort of more granular level for younger Earth's history. So I currently have a student who's working on sort of the Devonian through Carboniferous interval and testing hypotheses like those of Thayer's about the biological bulldozers. And again, the student who's just started to do some work in the Jurassic, but I think that there's a lot of work still to be done to sort of resolve the nature of the first order bioturbation records through this interval, and that will doubtless help us to interpret what these minor scale fluctuations might mean for the younger Phanerozoic. But regardless, even though that's very tantalizing and still a little bit inconclusive, I think that this is a very remarkable drop off that we see between the early Paleozoic and the younger intervals that I suspect will continue to hold up even as we add more data to this data set.
Excellent, very interesting, thank you.
All right, thank you. Yeah, Anna?
Thanks, Lydia. I really enjoyed your talk. I was wondering about the controls on bioturbation in the norm.
Mhm.
And if you could go back to that map figure and like, do you have an understanding of what needs the different depths in different places? And like, does this allow you to make predictions for the rock record or for future?
Yeah, that's also a great question. And the answer is that it's turned out to be pretty complicated. So previous studies have suggested that, for instance, sedimentation rate might be sort of a master variable. So some of Bernie Boudreau's work, for instance, has suggested that sedimentation rate may be a large driver, at least for the biodiffusion coefficient. And we've been doing this for both biodiffusion intensity and mixed layer depth. And others have suggested that that TOC would be sort of the rock equivalent, or the rate of organic carbon delivery to the seafloor may play a substantial role. And what we've found is that it's actually no single factor. So there's a combination of factors, including things like organic carbon availability, as well as water depth, which is itself correlative with a whole bunch of things that appear to be particularly strong predictors of mixed layer depth and biodiffusive mixing intensity, but it's not any individual factor. And it's even with the mixed layer depth ended up being really complicated. For the biodiffusion, there were sort of a few factors that came out more at the forefront, like the ones I mentioned. But yeah, there really isn't a single predictor variable, which makes things harder down the road. But you know, in some ways is not unsurprising. When you add biology to the mix, it always makes the patterns a little bit more complex, a little bit messier. Yes.
Really cool stuff on the England Van Capellen path
Thank you.
model.
Yeah.
But I had a question specifically, I really like that figure that showed the different factors,
Mhm.
reactive iron and race of sulfate reduction.
Yeah, and I think you mean this one, right? Yeah, so we actually subsequently, as part of the follow-up paper that we did was more on bioturbation, we looked at rates of oxide delivery to the seafloor.
Sure.
And I'm not certain if we looked at dissolved iron concentrations in the bottom water, so correlate. That could potentially be a really interesting one especially for thinking about episodes of deoxygenation, or for the Neoproterozoic and lower Paleozoic, for instance. But rates of oxide delivery to the seafloor, we could say J oxide, actually are also a very powerful lever on this. And I think, you know, that's one of the sort of avenues I'm really keen to explore next, thinking in particular about the lower Paleozoic and the persistence of ferruginous ocean conditions in a number of settings, how much of an impact that may have had. And then there was another parameter that you asked about, can you remind me, besides the oxide.
Race of sulfate reduction.
yeah.
My recollection is when we played with this for a while, and it was the role of iron oxyhydroxide trap.
Yeah,
And that if you have high rates of pyrite formation, you
Yeah.
lock up the iron, you may take that away and thus not lock up phosphorus and CFA.
yeah, that's a great question. I don't think we've looked at rates of sulfate reduction in this context, but that would be another really intriguing avenue to explore.
It's complicated
Yeah,
because it depends on a lot of different factors, but that's exactly what you're showing.
yes,
Cool.
indeed. Yeah,
While this is up, so I noticed on the right, you've got the phosphorus variable, CFA.
yes.
Did you also, and I think I know the answer, but did you try tracking total phosphorus variable? In other words,
Yes.
did you change, does it change the story much? Are there circumstances where total phosphorus actually might stay the same regardless, even if CFA is going down?
Yeah, so that's a great question. And we did in fact sort of keep track of all three of the CFA, the organic bound, and the oxide sorbed, as well as sort of how much got liberated in flux. So I think we do have mass balance there. Yeah, so within the sort of range of boundary conditions that we explored for these baseline models, always seen as though CFA were the strongest lever. But we actually performed a number of additional sensitivity analyses with not only the sort of ones we isolated for more detailed exploration were calcium, as well as the iron oxide flux and rates of organic carbon delivery. And then we also looked at sed rate. And sed rate is perhaps not surprisingly, as it is for many biogeochemical systems, can be a major lever as well. And what we found is that under sort of more extreme end members for sed rates, but still sort of commensurate with what we observe in marine settings today, though perhaps not so much continental margin settings, you can actually have organic bound phosphorus be a relatively more important phase. So the sort of distribution between the different phosphorus species can shift under different environmental boundary conditions, including sed rate.
So does it change the story then? Is calcium still the main player, or do you need another figure that shows sensitivity as a function of because of the different phosphorus?
Yeah, no, that's a good question. So for this study, we varied each of those in concert with biotubation, in particular with biodiffusion. And we also looked at biodiffusion with a sensitivity to bottom water oxygen as well in that framework. But what we did not do for this study, at least, and I'm trying to think back of whether or not we did these runs anyway, but for this study, we didn't look at calcium and sed rate together. We just looked at those with biotubation individually. But yeah, another interesting avenue. Mhm. Other questions?
Excellent. Thank you so much.
All right, thank you.